Today we're going to explore dihybrid crosses, and di means two. So di is looking at two different traits. So in this example, we are going to be looking at ear length. Big E means you have long ears, and little e means you have short ears. We're also going to look at foot odor, another characteristic or trait. Big F equals you have smelly feet, little f means you have sweet feet. Before we begin, we need to learn about FOIL. FOIL is a learning tool that we use to help us remember how to cross different traits. FOIL stands for first, outer, inner, last. And again, if this was one trait, so say for example, these two letters were ear length, these two letters would be foot odor. So first means we're going to cross the first letters together. That's first. Then we're going to do outer, so the outermost letters. We're then going to do inner, and then we're going to do the last. This is to determine the gametes from this cross. So for example, we have long and smelly are crossed with short and sweet. So again, long, if it's purebred long and smelly, it would look like this. And short and sweet would be lowercase. So first, outer, inner, and last. So you'll notice that all the combinations of these two are going to be big E, big F. All those combinations are going to be that. Let's look at shorter and sweet. So again, first, outer, inner, last. And again, all the combinations of the gametes from this parent are going to be little e and little f. So again, these are all the variations of this cross for this. These are all the possible gametes, either sperm and egg combinations that this parent can make. And again, these are all the sperm and egg contributions that this parent can make. So just like monohybrid crosses, we're going to put one parent on the top of the box, and we're going to put one parent on the side. However, in this case, we're going to put two letters. So I'm going to put this big E, big F here, and I'm going to put this little e and little f here. Just like monohybrid crosses, we put the capital letter first. However, we are going to organize the same letters together. So we're going to take this big E down here, this little e comes across, this big F here, this little f comes over. So you can see now we have a heterozygous offspring with big E, big F, little f. We're now going to do a heterozygous cross, but first we need to figure out what all the possible gamete possibilities are within one of these parents. Again, we need to apply FOIL. So I have big E, big F, little f. So my first is big E, big F. Outer, big E, little f. Inner, little e, big F. And last is little e, little f. Another way to look at it is that there's a pattern. Both of these are capitalized, both of these are lowercase, one of these is capitalized, and the other is lowercase. Again, lowercase, capitalized, they just switch places. Since these parents are the same, again, all the possible gametes for this one will be the same. So it's going to be big E, big F, big E, little f, little e, big F, little e, little f. Again, the pattern you can see, both capital, both lowercase, and these two switch off. This is big E here, big F here, lowercase f here, lowercase e here. Let's plug in these gametes into our giant Punnett square. We're going to do this parent first. So we have big E, big F, big E, little f, little e, big F, little e, little f. Likewise, let's plug this parent in over here. Big E, big F, big E, little f, little e, big F, little e, little f. Let's start to fill out the boxes. Again, we want to keep our E's and F's together. So this is big E, big E, big F, big F, big E, big E, big F, little f. Let's review our genotypes and phenotypes. Our genotypes are big E is long, little e is short, big F is smelly, little f is sweet. So our phenotypes, what our organism is going to look like is either long and smelly, long and sweet, short and smelly, and short and sweet. So let's first look for long and smelly. I'm going to highlight those in a purple highlighter. So long and smelly, it has to have at least one dominant trait. So this is long and smelly. As long as it has a capital E and a capital F, it is long and smelly. So these are all big E, big F, long and smelly. Big E, big F. Nope. Big E, big F. Nope. Big E, big F. Big E, big F. Nope, nope. Big E, big F. Nope, nope, nope. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9 long and smelly. Now let's look for long and sweet. I'm going to do that in blue. So again, to be sweet, it has to have two recessive alleles. So I have a big E, and both of those have to be lowercase f's. Big E, lowercase f. Nope, nope, big E, lowercase f. And for that, we have 3. And the last one, we are looking for short and smelly. So short has to be 2 recessive traits and a capital. So we have one here, we have one here, little e, big F. Okay, so that is my short and smelly. And I have 3. And that leaves me with 1 with both recessive traits with a 1. Whenever you cross a dihybrid cross, you always get a 9, 3, 3, 1 ratio. You'll always see that. Those are dominant traits, dominant recessive, and completely recessive traits. You always get a 9, 3, 3, 1 ratio.